Hello, my name is Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. In the fall semester, I teach my beloved microbiology, but also a freshman writing seminar. Now, these seminars are designed to help students learn how to carry out various functions that they have during the first year of college, how to write an essay, how to do group work, and I decided as a central part of this course to focus on symbiosis and parasitism. So I called this freshman writing seminar, Never Really Alone, Parasitism and Symbiosis Around and Within Us. And fortunately in this course, I'm able to have some very famous people Skype or televisit in and talk to my students. My students read some basic papers by those individuals. They come up with questions. I send those papers to the scientists in question, who then televisit us and answer student questions. And it works out very, very well. It's quite an honor for my students, in fact. This particular session focuses on Dr. Meghan Duffy of the University of Michigan, who studies the way that the small aquatic crustacean Daphnia deals with parasites. I hope that you enjoy this session as much as my students and I did. Thank you very much.
Oh, yeah, I know. So, folks, I would like to introduce all of you to Professor Meghan Duffy of the University of Michigan. First time I practiced this, I said Michigan State. That would be really bad to say. So I've trained myself carefully. And Professor Duffy is here to talk to you about some of her work involving parasitism as they relate to Daphnia and related questions. I have taken the questions that you sent me, collated them, sent them on to Professor Duffy, and I would like to introduce you all to her now. So what I'm going to do is turn it around so you don't have to look at my mug.
Okay.
Yeah, tell me about it. Wave, everybody. There you go.
Hi, everyone.
And so traditionally, Professor Duffy, we just let you take over from here.
Okay, sounds good. So thank you all for doing the readings for today. Dr. Martin sent me the list of questions that you came out with, which I've now annotated on my end. So I was just going to lead in by going into some of the questions that you had, unless someone there had a burning question they wanted to ask right at the start.
Burning questions? I think they're more tepid.
Yes, I figured. Okay, so I'll start in then. Several of you had questions that relate to phenotypic plasticity and maternal effects. So there were, I think, at least 4 or 5 questions that all related to that. So I thought that might be worth going into. So, okay, so phenotypic plasticity is when one genotype can have multiple phenotypes, right? So one set of genes can create an organism that has different traits. And several of the questions related, like I see one from Emerson here. Who's Emerson? Can I see Emerson? I think I can't, or at least no one's waving. There we go. Okay. Hi, Emerson. So you had a question about how phenotypic plasticity might affect the way Daphnia is affected by parasites, which is a great question. And that's something we have looked at. So for a while, we've known that Daphnia, one genotype of Daphnia can have different phenotypes depending on whether predators are in their environment. And so we know, for example, that if they have invertebrate predators in their environment, they sometimes grow bigger faster. Because these invertebrate predators, they're called gape limited, so they can only open their mouth a certain amount. And so if the Daphnia can grow so big that they can't fit in their mouth, that's good for the Daphnia. What we found is that bigger Daphnia are more susceptible to parasites. And so what we found is that, so we did this experiment where we were kind of mean to the Daphnia. So we tricked them into thinking that there were invertebrate predators in their environment. And then instead we exposed them to parasites. And we found that when they defend themselves against the invertebrate predator, they become more susceptible to the parasite. So these poor little Daphnia I think of as being kind of stuck. Like there are a lot of things out there that are trying to kill them. And there's no one strategy that lets them defend themselves against against all of the different things that are trying to kill them. Let me see, there were a few other, there was a question about how, that sort of relates to plasticity, which is that Daphnia, sort of how the environment influences what sex Daphnia are. So most Daphnia, when we go out to sample, are female Daphnia, but then at this time of year, they switch to reproducing sexually for the species we work on. And there was a question about whether it was related to temperature. It's actually this very cool system. So temperature cues or crowding cues or photoperiod cues, there are different cues that can trigger the shift. But Daphnia males are genetically identical to their moms. So it's actually just a hormonal signal that the Daphnia send to the developing embryos, and that makes them male. So that's kind of neat. And then once there are enough males around, then the female Daphnia become sexual, and then they produce these resting eggs. And this is one of the reasons, so there were some questions about, let's see, Heather had a question about how we can trace genetic adaptations over time. Who's Heather? Can you wave? Hi, Heather. And then someone else, now I'm not gonna be able to find it, had a question about, oh, Ross had a question about how I started working on water fleas, and these things actually all end up relating. So who's Ross? Say hi. Okay. So when I was an undergrad, so I went to Cornell, and my family is like not an academic family. So my father is retired now, but he was a New York City firefighter. My mother is a nurse. I knew I was interested in biology, but I didn't really know what I wanted to do beyond that, other than I knew I didn't want to go to med school. And so I was just taking different biology classes, and I took evolution, and I had this really amazing teaching assistant in my evolution class. And she based on that class, I got more interested in doing research, and she put me in touch with someone who did work in the lab. And the research I was doing with that person was on fish and speciation, and I thought fish seemed kind of cool. But then when I was in that lab, I started hearing about this other project that people were doing that related to Daphnia. And I had no idea what Daphnia were when I started in the lab. But after hearing more about them, I thought they sounded pretty neat. And one of the things that sounded the neatest about them is they have these, what we call egg banks. So these, when the Daphnia reproduce sexually, so to link back to that, the offspring that they make are enclosed in these dormant egg cases. And so it's basically like a seed for a Daphnia. And those seeds, which are called Aphipia, go and they settle down on the bottom of the lake, and they end up, some of them hatch out the next year, but a lot of them just get stuck in the mud. And so over centuries, then you build up these genetic archives going back in time in the sediment. So you have, let's see if I can get my hand in the picture, right? So at the top of the sediment, you have the newest eggs that are formed, and then as you go back in time, the deeper you go in the sediment, the older the eggs are. So this is neat because then you can hatch out these things from up here, and those are recent, and you can hatch out some from, say, a few decades back, and then you can hatch out some that are even older, and you can use that to look at how a population has changed over time. So that was pretty amazing to me when I learned about that as a student. Like you have this naturally occurring genetic archive in every lake and pond, which was pretty, I thought that was really neat. And that's what really got me hooked on this system. So there's a whole field that they call resurrection ecology, where you take these sediment cores and then you hatch out Daphnia from different depths. They can be other animals, but a lot of it's with Daphnia. And then you can look at how different traits of theirs evolved over time. So one study that was done looked at how their ability to graze on toxic cyanobacteria changed over time as the lake got polluted and had more cyanobacteria in it. Someone who works in my lab, she did work recently before she joined my lab, looking at how Daphnia evolved in response to cadmium and copper pollution in their environment. And you can do that by going back in using these egg banks, which is a really neat aspect of this system, and it's something we would really like to do more work with, looking at tracking how they evolve to parasites using that egg bank. These studies end up being really hard to do because you have to, it's not trivial to hatch out the Daphnia from all those different ages and then to hatch out parasites too. There's one person in Europe who did that and got a paper in Nature, which is one of the top 2 journals based on that work. So there has been some work done on that. And she was able to show that they were adapting, that both the host and the parasite were evolving in response to each other. So I'll pause there for a second because I've just done a lot of talking, and I'll see if anyone has follow-up questions about any of that.
How long can Daphnia eggs survive?
Yeah, that's a great question. So we know that over time they, you get fewer and fewer of them hatching, so they are dying. But people have been able to hatch eggs that are hundreds of years old. So there was one study recent, which is pretty amazing, right? Like, so there was one study that was done recently where they were able to hatch a few genotypes that were very, very old. They couldn't actually quantify how old they were because the way we date these cores is we use lead-210. And they were able to hatch some from so far back that lead-210 is no longer a useful isotope for measuring. So that's very old. So all they could say was they were hatching ones from before European settlement. This was a study that was done in the Midwestern US. So I think the oldest one where they've dated it well was, the oldest one I can think of, I think was a copepod, and it was about 400 years old, a bit old, over 400 years old.
See, this,
so they can be very old.
This interests me, Professor Duffy, because as you know, I have this unusual fixation on tardigrades.
Yes.
And everyone, well, unusual is not the right word, perhaps odd. Anyway, the point is that everyone says how long that they can survive when they're in cryptobiosis. And I don't really know what the answer is. I think for tardigrades, we know about 35, 40 years because of some Arctic samples. But here we have stuff even older.
Yes, yeah, so they can be really old, which is, so again, we know they're dying at some level in this stage because you can get lower and lower proportions of them hatching the older you go, the further back in time that you go, but they can hatch some very, very old ones, which is pretty neat. Yeah, and this is true for both, so Daphnia, or what I've been talking about, they're what I focus my research on. So Daphnia, you read about this, they're a model system, and that's part of why I work on them, but they're also just very important ecologically. So in lakes and ponds, they're the dominant grazer. They're the dominant thing that's eating the algae, and then fish they're a dominant source of food for fish. So they're a key link in these lake and pond food webs. But there are also other zooplankton out there. So things like copepods. Copepods are a really dominant zooplankton in oceans. They also can make these resting stages. And then rotifers are a smaller zooplankton. They also make these resting stages. So it's a pretty common strategy. And so it means that you have these egg banks of all of them in the sediments. So you may be familiar with some work that people do on bacteria. They put their bacteria in the -80 freezer to save it so that then later they can take them out and they can compare a later population with an earlier one. This is like a natural -80 that happens out in lakes, which is pretty, like I said, that's what got me hooked on the system. Okay, are there other follow-up questions on that? Okay. So there was a question about, well, there's one question that related to how climate change might influence these ecosystems and might influence the host-parasite interactions. And that is something that's something that we're interested in. So it's not maybe, I don't know, 5 or definitely 10 years ago, if you asked me if my work related to climate change, I would have probably said no, but that's something that's emerged over the past few years. So one thing we're looking at is how lake color affects disease. Lake color doesn't sound like the most exciting topic, but so lakes are getting browner because of global climate change. So there are more rain events, more storm events, and that brings more material into lakes. That includes dissolved organic carbon, which is basically what I'm drinking here. So my tea, right? That has a lot of organic material that's made the water in my mug dark. That's what happens in lakes too. And that makes it so that light doesn't penetrate as far into these lakes, which means that UV doesn't penetrate as far into the lakes. And we've shown in my lab and in collaborations with some other people that several of the major parasites of Daphnia are really sensitive to UV. We don't know why. We thought Daphnia were really sensitive to UV, but it turns out the Daphnia parasites seem to be even more sensitive. And so one thing we're looking at is whether global change then will increase disease in Daphnia populations and whether it might shift the communities of parasites that we see in our Daphnia populations. And then also related to that is work that a grad student, Crystal Sanchez, is doing in my lab, where she is looking at how the type of food Daphnia eat might influence whether they get sick. So we don't really think of animals other than humans as medicating themselves, but there actually is some evidence that this happens. So with monarch butterflies get infected by a protist. And what they found is that if they feed on milkweeds that have more of these toxic chemicals, so have you learned about milkweeds in any of your classes? So milkweeds contain these chemical compounds called cardenolides. And that those are toxic to vertebrates. So you would not want to eat a milkweed. You would feel very, very bad if you ate a milkweed. But how bad you would feel would depend on which species of milkweed you ate. Some of them don't have as much of this cardenolide as others. And what they found is that these cardenolides protect the monarch butterflies, which live on the milkweed, against their parasites. And so a mom monarch butterfly can sense if there are parasites around, and then she goes and lays her eggs on a more toxic species of milkweed, which then helps protect her offspring. And they found that Drosophila, so we're finding now that different animals do this, so it's really self-medication or they're medicating their offspring. So like I have a baby who I gave medication last week for a fever. It's sort of like these monarch butterflies are doing the same thing for their babies, which is kind of neat. So all of the work that's been done on that has been in terrestrial systems, and Crystal, my grad student, is interested in whether some of this might be going on in aquatic systems. So she's looking at whether Microcystis, which is a really common species of cyanobacterium, so it's a species that blooms a lot. So if you hear a story in the news saying that there's a toxic algal bloom in a lake, there's a very good chance it's Microcystis. So it's the one that bloomed in Lake Erie and made Toledo have to shut down its water supply a couple of years ago. It's been blooming in Florida. It's a very common problematic nuisance cyanobacterium. And some strains of Microcystis produce microcystin, which is a liver toxin. And for a long time, we thought that, so aquatic ecologists were looking at whether microcystin is something that is affecting the grazers, so the things like Daphnia that are eating the Microcystis. But there's actually not a lot of great evidence that it is. And then there's a more recent paper that shows that Microcystis has been making this toxin since before Daphnia or any other animals existed. So that suggests that it at least didn't evolve initially to make this toxin as a defense against herbivory. So what Crystal is looking at is whether it's a defense against other microbes, and if so, can the Daphnia sort of medicate themselves? If they eat some of this microcystis when they're exposed to parasites, does that help protect them? And she's finding that yes, it does help protect them, which is pretty neat. And then again, has links with climate change because we have more of these blooms of these toxic, we might have more of these blooms of these toxic cyanobacteria under climate change scenarios. And so that again then might have impacts for how much disease there is in the Daphnia. Okay, I'll pause again for questions to see if there's anything related to that. Probably none of you have thought about things other than humans medicating themselves before, right? I think that's kind of a neat angle on this stuff.
No, I knocked. That was my fault. I was so surprised by the idea.
You're so surprised that they're medicating themselves.
That's right.
Yes. Okay, so there was another question about whether Daphnia have antibodies that are protecting them against diseases. And part of why I thought this question was interesting is this is something that people first looked at in the 1880s. I'm gonna get seasick if I keep spinning around like that. So there was someone called Ilya Mechnikov who worked on Daphnia parasite interactions in the 1880s. So he has a few papers that are really important, and this was before English was the generally used language for scientific publications. And so these papers are in Russian and French and German, which is a struggle for me because I don't speak any of those. But he has these papers where he's describing, he's looking at different things. So one of them, he's just interested in how a parasite in Daphnia looks different over time. And it's clearly a very different time. It was clearly a different time for doing science because he would go to a pond and he'd collect a Daphnia and he'd put her on a microscope slide and he'd just leave her there and he'd come back every few hours and he'd record what he saw, and that was a paper. We don't get papers out of that anymore, but he did really important foundational work on this. But one of the things he saw when he had his Daphnia just hanging out on the slide, I'm not exactly sure how he kept them alive for so long, but somehow he was able to keep them alive. He saw a Daphnia infected with this fungal parasite that we work on a lot in my lab, which is now called Mechnikowia after him. He watched a Daphnia who had these Mechnikowia spores in her gut, and he watched one of the spores puncture the gut wall and shoot the ascus puncture the Daphnia gut wall and shoot the spores in. So that's how they get infected. And then he watched a host cell come along and engulf that parasite cell and wall it off. And that was the first time anyone had ever seen a host cell attack and effectively wall off a foreign particle within a host. So that was the first evidence ever for first time someone watched a host immune system at work, which is pretty neat that it was in this system that I work on. And it makes it interesting that there's been very little work done on host immune responses in Daphnia since it's the original host immune response that was seen. There's someone who did his whole thesis on Daphnia immune responses to a bacterial parasite. But what he found is that the immune response seems to be more of a symptom of infection than an effective protection against infection. So genotypes that mounted a very strong immune response were the genotypes that got very heavily infected. So it didn't seem to be effective, and it's not really clear. It's not clear then why they're mounting that immune response. So there's a lot to be done there, and we still don't know very much about it. I'm starting to get some feedback on my end. Let me try changing my volume settings and see if that, okay, that's a little better. Can you still hear me there?
Absolutely.
Okay, it's distracting when I hear myself echoing. And that also links to another question on why it took so long for Daphnia to be researched. So it's interesting because Daphnia had been studied for a long time. There are lots of studies of them from the 1800s. And Daphnia parasites were also initially studied in the 1800s, but then there was a long time where basically no one was doing work on them. And I'm not sure why. I'm not exactly sure why people sort of forgot about Daphnia parasites. But then there's someone, Peter Ebert, who is in Switzerland, and he has been, he's really led this re-emergence of work on Daphnia and their parasites, and he's the one who wrote that perspective piece that you read. He has done almost all of his work in swans, and so it took a while longer for people to realize that there were parasites attacking Daphnia that were living in lakes. And some of the initial work on this was done by my PhD advisor, Alan Tessier, and it was not something he was setting out to study. He just was going out to collect Daphnia, and he noticed at one point that they all looked funny, and he looked under a microscope and could tell that they were infected. And then he went back to that lake a few days later because he thought, well, this is neat, let me try to collect this parasite, and the population had crashed. And so that was the first sign that maybe these parasites were important and we were just missing them. And so my theory on why people study Daphnia in lakes a lot. It's like a really major topic of study. But again, people were missing these parasites. And so my theories on why that happens why that happened is that the parasite epidemics tend to be mostly in the autumn. So my lab's field season just ended on Friday. So we always sample from mid-July through mid-November. And we always stop. Today is the last day we can possibly sample because November 15th is the start of deer gun hunting season. So it's no longer safe for us to be out sampling these lakes. So we always stop by November 14th. And so it's late. Usually people who are studying Daphnia in lakes would be sampling over the summer, and they wouldn't be sampling still at this time of the year. And then the other reason why I think they got overlooked for so long is that if you preserve your Daphnia in ethanol or formalin, the normal things that we preserve Daphnia in, you can no longer tell that they're infected. So I think it was the traditional way people would sample would be they'd go out, they'd collect a bunch of animals, they'd preserve them, and they'd count them a few months later. And since they weren't looking at them under a microscope right away, then they were missing all of these parasites. So people, even if they were sampling at the right time, they weren't sampling in a way that would let them see the parasites. So, and even now, I think we're probably missing a lot of parasites. I think we're probably missing a lot of viruses because we would have to sample a very different way to detect viruses, and we mostly aren't doing that. There are a couple of people looking at viruses, but very, very few. Okay, follow-up questions on any of that stuff?
Natalie has one.
Yeah, I was wondering, in the essay that we read, it talked about the maternal effects, and I was just wondering what kind of maternal effects help protect Daphnia from diseases.
Yeah, so what kind of maternal effects help protect Daphnia from diseases? Is that your question? Yeah. So there was one study that found, so they used a bacterial parasite where there are very strong differences between genotypes in whether they can infect a host. And what they found is that if they infected if a Daphnia was infected with one genotype, and then they cured her of her infection by giving her antibiotics, which I always think is fun, like that we can give our Daphnia antibiotics. They cured her, then she had more offspring. What they found is that those offspring were less susceptible to that particular strain of the bacterium that the mom had been infected with. So it wasn't just that the mom was transferring something that made them less susceptible to all bacteria, but there was something that was making them less susceptible to that particular strain of the bacterium, which is pretty cool, but we don't know what it was. So we don't know at this point what we know. In some cases, it might be something about like how quickly the offspring grow, but there are other things going on that we don't know yet. So there might be epigenetic changes. There might be changes in the sorts of nutrients that certain compounds that she sends to her eggs, but we don't know yet what they are. So that's a great question that we can't fully answer yet.
So this fascinates the you-know-what out of me, because one of the things that I teach about in my microbiology class is the difference between innate and adaptive immunity.
Yeah.
These preset innate systems are what I would expect to see in invertebrates like Daphnia. But what I'm smelling is something adaptive in this case because of the specificity.
Yeah. Yeah, so it's one, I agree. So as I was saying before, so this first evidence of immunity that Metchnikoff found, right? So that's certainly innate immunity, where it's just how many phagocytes are floating around in the hemolymph, which is Daphnia version of blood, to wall things off. This does suggest that there's some sort of adaptive immunity. We have no idea what it is. And it's the sort of thing where it would be nice if people replicated it to see how many species can do this. Can they do it to different genotypes and species of the parasite and all that? But yeah, it does. That result definitely suggests some form of adaptive immunity, which is pretty cool. We don't normally give invertebrates credit for that sort of thing. You were saying that, another thing popped into my head. Oh, I know. So there is one there. So this person, Dieter Ebert in Europe, his lab has done a lot of work just trying to understand what allows, what determines whether a Daphnia gets infected or not. And he's found for this bacterial parasite and Daphnia, it seems to be a locking key sort of mechanism where the spore attaches to the esophagus of the Daphnia, and certain spores can attach, and certain spore genotypes can attach, and certain spore genotypes cannot attach. And that depends on the host and the parasite genotype. We found in my lab and in work with collaborators that there are a lot of things related to feeding that influence susceptibility then. And so we know most of these parasites infect via the gut. So things that affect how quickly food passes through the gut or how it packs in the gut seem to influence whether they get infected. Which also then links with the work I was talking about before, where some of these toxins which come in with the food, that a lot of that interaction is probably happening in the gut as well. So these Daphnia, I guess several of you looked a little startled when I pointed out that they get infected by having their gut wall punctured, which is a little, it's sad for the Daphnia. I should also say that at the end of infection, so these Daphnia don't transmit disease by, like, it's not like they're sneezing on each other. At the end of infection, for most of the parasites, what happens is they eventually die from the infection, and then the corpse, the Daphnia corpse, contains all of the spores within it, and then when that ruptures, all of the spores get released. And so this is something, one of the things we worked on is how these invertebrate predators can influence that, because the invertebrate predators eat the Daphnia and then regurgitate the infected corpse, which we're starting to sound a little bit like a zombie movie here. They regurgitate the infected corpse and all of those spores, and that helps spread disease. So it's a little gory sometimes out there in the plankton. It's all happening at a very small scale that we can't see, but there are a lot of things attacking each other in a way that's a little gory if you scale it up to human size. Are there questions about any of that?
One of the things that we've talked about a lot, just in background for this course, is intermediate hosts that parasites have. Do we have any involvement in Daphnia with that?
No, these all seem to be directly transmitted. Well, they're not strictly speaking directly transmitted, because that would be sneezing on each other, right? They're environmentally transmitted, but as far as we know, it's one Daphnia host releases spores into the environment, and those get taken up by another Daphnia host. So there doesn't seem to be another host, like a vertebrate host or snail. Snails always seem to play a role in those complex life cycle aquatic parasites. Those don't seem to be playing a role here. So that actually reminds me, there was another question, or there were two other questions I wanted to get to. One was asking about whether humans can sometimes be, it was talking about how some of the reading had talked about how chemicals can be important in influencing host-parasite interactions. And it was asking about whether humans might potentially be manipulated by chemicals related to parasitism, which links to these complex life cycle parasites. So when I first started working on these parasites, I was a little disappointed that they weren't getting transmitted to something else, because those are the cases where you see these really neat behavioral modifications where a parasite, so one where this has been really well studied is with Toxoplasma, where it's a parasite that is usually, it spends a lot of time in rodents, so in things like mice and rats, and then, but then it needs to get into cats to complete its life cycle. And so it makes it so that the mice are actually attracted to the rats, or sorry, mice are attracted to the cats. So they like are attracted to their predator, and that's because this parasite is manipulating the brain of the host. And they've actually shown that what it manipulates is the region of the brain that controls sexual attraction, which is kind of weird. So then they're attracted to their predator, which then can eat them, and then the parasite can complete its life cycle. So Toxoplasma is a parasite that can get into humans. It's the reason that pregnant women are told not to change cat litter boxes. That's the concern, is that that parasite might get from the cat litter into a human. And so there has been one study that looked at rates of Toxoplasma infection at the country level, and then at country level measures of neuroticism, which one thing that was interesting to me about the study is that there are country level measures of neuroticism. But so how neurotic people are in a country. Because we know this parasite is manipulating the brain, it changes things like aggression, and there is a positive correlation. And when I teach about this sometimes in like disease ecology classes, we then go into how correlation is not causation and all that. But it is an interesting correlation where it does seem like some parasites that affect humans might be manipulating our behavior through making changes in the brain. Yeah. Okay, the other question that I just noticed that I want to make sure we have time to get to was about how I became a professional scientist, and then what are the important things for preparing and applying for grad school. So I would say the most important thing, and I say this largely based on having spent a few years on a grad admissions committee, is to get research experience as an undergrad. So one thing that some of the grad applications that I found the most challenging to address were from students, or to assess, were from students who had a good academic background, but didn't have any research experience. because there are differences between enjoying being a student and enjoying doing research. They do often go together, but some people like being a student and they don't like doing research, and vice versa. So there are programs where you can get research experience over the summer called REU programs, so research experience for undergraduates. And those are things that you would apply for usually in January and February, and then you would do them over the summer and you would get paid to do research somewhere. So it's, those are really great programs to apply for, and getting research experience is really, really, really useful if you think you're interested in going to grad school. Are there questions related to going to grad school, scientific careers, those sorts of things?
Some of our students are thinking about that. Some aren't. These are in their first semester, these students, okay, of their time here. And we often
talk about— Which is not too early to be thinking about this.
And this is something that I have, we have our own summer research program here, and I've been battling with them for some years about getting people started early. Mm-hmm. And I think your idea about REUs are very important. We have a program here, and there are some, like the American Society for Microbiology has a good way of supporting student work at the host institution, and I'm sure ecological societies have the same thing going, but you're absolutely right.
Yeah, and it's something, if you ask me my first semester in college what I wanted to do, I would not have said become a professor, and I would not have said do research. I think I would have had a very vague, uncertain answer. So I didn't, I think research seemed interesting to me. I also came, when I got to college, I realized how much better prepared many of my classmates were than I was. And that was a little overwhelming, and it made me think at first that maybe I learned that some of my classmates in college had been doing scientific research as high school students, and I was like, well, I guess I'm too late. Like, I just initially thought like, oh, well, I'm behind. If they were already doing it, it's too late. And then it was, as I said, it was when I was a sophomore, I took a few classes that really captured my interest, and it was at the end of my second semester of my sophomore year that I decided I was interested in maybe doing some research. And so I didn't start really doing research until the beginning of my junior year in college. So it's good to start early, but if it takes you a few years to figure out what you want to do, that's also fine. There's still time to get research experience and to do research.
You have to allow the parasite time to infect your brain to direct your behavior, right?
Yes, exactly, exactly. We unfortunately don't have funding for it anymore, but we used to run an REU program here that was specifically aimed at students who had just finished their first year in college, because the idea was we were trying to recruit students to ecology and evolutionary biology, and if a student doesn't decide they're interested in ecology and evolution until their senior year in college, it's still possible for them to have a career in it, but it's a little harder than if they did that in their first or second year in college. So getting experience early can be useful. Well, one reason it can be useful is that if you don't like it, it still leaves you time to explore other options. But if you do like it, it gives you more time to build on things. All right, I see that we're getting close to your 1:40, or sorry, it's 1:45 here, your 10:45 time there. Are there any last questions that people have?
So this is a good opportunity, folks, because I've been following Professor Duffy for some time. And there is very little about this kind of subject she doesn't know. You might think it's just about Daphnia. She knows a lot of stuff outside Daphnia, especially involving parasites and symbiosis. Mm-hmm. So any last minute questions that any of you might have?
Yeah.
Maggie.
We recently had like a podcast assignment where we listened to a podcast about like parasitism.
I can't hear very well. I heard you say podcast, and that's about it.
I listened to a podcast recently, and they discussed a study done about how individuals involved in automobile accidents, with like drivers and pedestrians, were found to have toxoplasma, like higher rates of toxoplasma. And I was wondering if you like thought that was a little far-fetched. Because it was. What was
the last part of it? So what I heard is that you listened to a podcast that found that people who'd been involved in automobile accidents had higher levels of toxoplasma. Is that right?
Yeah, and I was wondering, they were suggesting that it was possible the toxoplasma was making people more fearless, I guess.
Yeah, I have heard some studies about that. Yeah, where there is a thought that it might be affecting people's behaviors on like in a way that influences their daily risk taking. I'd believe it. So there was just a video that was circulating on Twitter where it was a rat, and the caption was something like, you know, this rat doesn't care anymore or something, but it was a rat that was running and like attacking cats. And my first thought was, that's a rat with toxoplasmosis. Like,
oh yeah.
It really can change. It certainly changes rat and mouse behavior a lot. So it wouldn't surprise me if it can affect human behavior, but I think we're still in early days of actual data on that.
The one thing that's fascinating to me is how insects are like little robots, and you could understand flipping this switch or that switch can have changes in behavior. But I expect that when it happens to humans, we have excellent justifications for why we're doing what the parasite says. Don't you imagine? I heard a story. I've told the students, I heard a story about someone who lived through rabies, and they asked the person why they wanted to bite people. And they said it just seemed like a good idea at the time.
Huh.
And I think there's some truth to that.
So wait, so they survived a rabies infection?
Yeah, very, very rarely in the there were a couple of cases in the 30s and 40s where someone came through.
Huh.
And it's very rare, not the more recent case where they use kind of a medical coma.
Right.
And it's meant, I don't mean to sound flippant about it, it's really terrible, but what I'm trying to get at is that when those switches get thrown in our brains, we probably justify them.
Yes. Yep.
And the problem is now we look at everything that way, especially when you think about the fact that in France, I believe 80% of the population has been exposed to toxo in one report that I saw.
Huh, that's interesting.
And they're not, I wouldn't call them super risky. They seem to drink a lot of wine and eat cheese and live a pretty nice life, right?
Right, they don't seem like a very aggressive population.
No.
Huh,
So any last questions for Professor Duffy? Well, let's give her a big round of applause for taking the time to chat with us. So while I shut down, please grab yourself whatever you want. You have something to give to me that you can leave up front with me, and I will see you all on Wednesday. Thank you so much. And thank you. I really appreciate you spending some time chatting with us.
Yeah, no problem. All right. Have a good day.
I'll talk to you soon.